So the chromosomal theory of inheritance, so by 1902, the chromosome movement during meiosis had been worked out. So Walter Sutton and Theodore Bowry, right? Sutton and Bowry noted that the behavior of chromosomes was parallel to the behavior of genes, and used chromosome movement, right? So Sutton and Bowry, so to explain Mendel's law, okay. And then there is like the meiosis is reduction division, equational division is, that is mitosis, right? And the two alleles of a gene pair, are located on homologous sites on homologous chromosomes, right? And the behavior of the chromosomes and the genes, that is almost parallel. The movement during the during what during like the okay, so during the meiosis. Now th Morgan, what about the th Morgan? Okay, the Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. So Sutton and Boveri both, but it's written that Sutton united the knowledge of, okay, but they both are like Sutton and Boveri give the chromosomal theory of inheritance. And now experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to the discovering the basis for the variation that sexual that that sexual अबे गिर जाएगा. uh, okay, so what about like okay, so the basis for the variation that sexual reproduction produce, okay. The Morgan work with like tiny fruit flies, that is Drosophila melanogaster, okay. And they show the sexual dimorphism, that is male and female flies are easily distinguishable. And now the linkage and recombination, the Morgan carried out several dihybrid processes in Drosophila to study genes that were sex linked, right? Now the processes were similar to the dihybrid process carried out by Mendel in peas, okay. Now Morgan hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males, intercaused their F1 progeny. Now he observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the like 9:3:3:1 ratio that is expected when the two genes are independent, right? So it was deviated, okay, from actual Mendelian dihybrid cross ratio. Now Morgan and his group knew that the genes were located on the X chromosome. The two genes in a dihybrid were situated on the same chromosome. The proportion of the parental gene combinations were much higher than the non-parental type. Now Morgan attributes this the physical association or the linkage of the two genes and coined a term that is linkage to describe this physical association of genes on a chromosome. The term recombination to describe the generation of non-parental gene combination. So recombination means like non-parental gene combinations, okay, on the same chromosome. Morgan and his group also found that even when the genes were grouped on the same chromosome, some genes were Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, that is show very low recombination, and while other very loosely linked, that is showed higher recombination. It means like if they are far apart, okay. And so example like white, that the genes white and yellow were tightly linked, so showed 1.3 recombination, while white and miniature show like maximum recombination that 37.2%, okay. Now his student Alfred Stewart would use the frequency of recombination between the gene pairs on the same chromosome as a measure of the distance between the genes and map their position on the chromosomes, okay. So today we use it in the human genome sequencing project, okay. Now in here like you can see like so what you are going to see here like okay. Okay, the strength of the linkage between Y and W is higher than W and M. So than W and M, that is what that is white and miniature, right? So white and miniature, and yellow and white. The strength, okay, strength is higher means the linkage is higher, so it has a low recombination, that is 1.3% of the recombination. And then white and miniature, that is 30.2 like percentage of the recombination. So that's it. Now the polygenic inheritance. So what about the polygenic inheritance, like human skin color and human height, it tells. And also, okay, so such traits are generally controlled by three or more genes and thus called as polygenic traits. So three or more genes, right? Like B B, capital C C. So we'll have the darkest skin color, then recessive, small a, small b, small b, small c, small c. We'll have like the lightest skin color, and intermediate has the like medium. Okay, intermediate skin color. If three dominant alleles and three recessive alleles. So this is it. Like now, in this manner, like we see the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. Okay, so this is now the pleiotropy. So as you can see in the pleioid, the effect of a gene on a single phenotype or trait. The effect of a gene on a single phenotype or trait. But there are some, like a single gene can exhibit multiple phenotypic expressions. Such genes are, these are the pleiotropic gene. And so example is the phenylketonuria. Okay, the disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase, that is single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction of in hair and skin pigmentation. So multiple phenotypic characters are being affected by a single gene. Okay, now the sex determination. So Henking gave the X body. Okay, now there are three types of sex determination, and that is XO, XY, and ZW. So XO mostly like the grasshoppers. Okay, and XY, that is in humans and Drosophila, and ZW, that is domesticated fowl or birds, that is chickens. So in first case, like XO, a large number of insects show XO, and all eggs bear an additional X chromosome besides the other chromosomes. The autosome, on the other hand, some of the sperms bear the X chromosome, whereas some do not. So all the eggs bear an additional X chromosome, that is XX, but in male it is like X and O. So one chromosome is absent, right? Now you can see and this is the male heterogamety. Okay, you see, due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosomes were named as. So okay, so grasshopper is an XO type of sex determination in which males have only one X chromosome besides the autosomes, whereas females have a pair of X chromosomes. So females have the pairs of X chromosomes, right? Now XY type of sex determination, in a number of other insects. Now large insects like the XO, but in other insects and mammals, including the man, the XY type of sex determination is seen, where both male and female have the same number of chromosomes, which is not in the case of like grasshopper. As we see, one chromosome is absent. So among the males, having X chromosome is present, but its counterpart is distinctly smaller. So Y is very smaller, and females have a pair of like X chromosomes. So both males and females bear the same number of autosomes, right? As you can see, like Drosophila, the males have one X and one Y, whereas females have pair of X chromosomes. Okay, now as you can see, like two types of sex determination, that is X1, XY, right? So now in first case, like either with or without X chromosome. So either with or without the X chromosome, or and some gametes with X chromosome and some with Y chromosomes. So either with, that is OO, and if it is not, like XO, and some gametes with X chromosome and some with Y chromosome. So X, XX, and XY, right? So such type of sex determination is known to be the male heterogamety, right? That male determines the sex of the progeny. Now the total number of chromosomes is same in both the males and females, but two different types of chromosomes are produced by the female. So this is female heterogamety. So this is uh, the two different sex chromosomes of a female bird has been designated to be the ZW chromosomes, and in this organism the females have one Z and one W chromosomes, whereas males have a pair of Z chromosomes besides the autosomes. So as you can see in that, so determination of sex by chromosomal differences both in humans and Drosophila. The female has a pair of XX chromosomes, homogametic. Females are homogametic, and the male, that is XY, that is heterogametic compositions. In birds, female has a pair of dissimilar chromosomes, that ZW, and male having like two similar ZZ chromosomes, right? Now the sex determination in humans, right? As you can see, the 23 pairs of chromosomes present, so total 26 chromosomes a individual have, like us. So 22 pairs are exactly same in both males and females, so these are autosomes, right? So one pair of X chromosomes present in female, and whereas the presence of X and Y, that is male gametes, right? Now come to the point, like always there is 50% of chance being like daughter or daughter or son, and the females are blamed, but the where is that line, where is it? Okay, it is even that the genetic makeup of the sperm that determines the sex of the child. Now sex determination in honeybee, as you see, female is diploid, and male honeybee, that is males, I think drones, these are haploid. Okay, so the sex determination in honeybee is based on the number of sets of the chromosomes an individual receives. So if it is union, that is sperm and egg, it can be female, and female can be a queen and worker, and unfertilized egg develops as male, that is drone, means of the parthenogenesis, that is formation of the organism from a without the fertilization, right? So that means the males have like half the number of chromosomes than that of a female, and the female are diploid having 32 chromosomes, and males are haploid, so having 60 chromosomes, right? This is called as haplodiploid sex determination system. And these males are produce sperms by mitosis. Okay, they do not have father and thus cannot have sons, but have grandfather and can have grandsons. Okay, now mutations.